Lincoln University PhD student Michelle Thompson is trying to find out if some grapevines are better than others at naturally defending themselves from a disease-spreading pest. She's conducting trials at Plant and Food Research, now part of the new Bioeconomy Science Institute. Cosmo Kentish-Barnes dropped into the lab to find out more.
Right now we're in the room with the equipment called the electropenetrography setup, also called EPG. And what we're doing here is measuring electrical signals produced by insects feeding on leaves, and we can interpret those signals to see what behaviours those insects are doing.
And the insects are tiny mealybugs.
They are. So I'm working with Citrophilus mealybugs. Citrophilus mealybugs are able to feed on lots of different parts of grapevines. They feed on the trunks, canes, leaves, berries, and these guys even feed on the roots. And they are tiny. They're quite a challenge to work with.
And, Michelle, they carry a virus.
They do. So mealybugs transmit a virus called grapevine leaf roll associated virus 3. That virus is a major challenge for the New Zealand wine industry and causes a disease called grapevine leaf roll disease, and that impacts the health of the vine. It reduces yields and it changes the grape quality as well. And that's estimated to cost millions of dollars to the New Zealand wine industry. It's more of a problem in red-berried varieties. So you think of things like your Merlot, for example, and in white-berried varieties like Sauvignon Blanc, even though the visual symptoms aren't obvious, it's still a big problem. And that's partly because during feeding, the mealybugs produce honeydew, and the production of honeydew can lead to the development of black sooty mould. And black sooty mould can impact the quality of the berries and ultimately the quality of the wine. So this is a major problem for wine growers.
Gosh, a huge problem. And currently, how are grape growers managing this pest and the virus it carries?
So current conventional management practices for dealing with grapevine leaf roll disease is to use chemical insecticides to manage mealybug populations in the vineyards, and also removal of virus-infected plants. And also, when removing vines, replanting of vines in the same spot can lead to the mealybugs moving onto those newly planted vines and feeding on them, and then they just reintroduce the virus. So managing those mealybug populations through use of insecticides is really key, and it would be great to have a more natural way of managing mealybugs and protecting our vineyards, something that's more environmentally sustainable and also less costly.
How did you come up with the idea to do this research with the mealybugs?
So electropenetrography as a technique has been around for a wee while. It's currently the only technique available to measure the feeding behaviours of sap-sucking insects. So sap-sucking insects being mealybugs and other things like aphids. It's been used a lot in aphid research. And because we want to be able to measure the feeding behaviours, not just see whether the insect lives or dies or whether it grows, and another way that you can measure success on a plant is looking at its ability to produce babies, for example. But we want to actually measure feeding, and this is currently the only technique available to do that. There isn't anyone else using this technique in New Zealand that I'm aware of. And so I'm very lucky I'm supported by some experts in the US who helped me learn the technique, trained me in how to do this. And so, yeah, it's definitely used in other laboratories around the world and is the standard way to measure feeding of sap feeders.
Can you describe what a mealybug looks like?
Yeah, absolutely. So mealybugs have 4 different life stages. They all start as a nymph or a crawler, which is about 0.8 millimetres in size. And then the females go through another 3 life stages, and they sort of look like a woodlice in shape. That's probably the best way I can describe them. The males look very different. So the males are quite a lot smaller. They have wings that have a bluish colour to them, and I'm not using males for this part of my work because the males don't feed. So they have quite a different purpose in life. So the females will feed, they transmit the virus, and the males do not.
Fascinating.
Yeah.
Now we are standing in front of 2 metal cages that are about 1/2 metre wide and high, and inside them are 4 wired up grapevines.
Yes, so the experiments that I'm doing involve working inside a Faraday cage, and what the Faraday cage does is it protects the system from other sources of electrical noise, because I'm trying to measure electrical signals, and these signals are very tiny. They're signals produced by a very tiny insect. And so what we've got here is we've got the electrical circuit involves, first of all, the plant. So there's a potted vine. There is a plant electrode that's inserted into the potting mix of the vine. A very tiny voltage is applied to the plant. A single mealybug is then attached to an insect electrode that's connected to an amplifier. And then what happens is when the insect inserts its mouth parts, which are also called a stylet, into the leaf, in this case they're feeding on the leaf, then that completes the electrical signals, and the signals can be recorded and then analysed.
How do you attach a wire to such a small insect?
Yeah, it's definitely tricky, and it took me quite a few tries to get it right. First of all, mealybugs produce a waxy coating on their cuticle, and so in order to get a good electrical connection, the first thing I need to do is brush off that wax. And I do that just using a very, very soft, fine paintbrush. So brush off the wax, which is white in colour, and underneath that actually shiny and brown. Then I'm connecting them to a very, very fine gold wire. So the gold wire is 18 micrometres in diameter, which is about 1/3 of the diameter of a human hair. So it's very light, and it doesn't impact the movement and the behaviour of the insect. That's important. We want them to be comfortable and behave normally. I make a really small loop on the end of that gold wire, and that's just so that the loop can sit on the surface of the insect. It improves connection, improves electrical connection between the wire and the insect, and also just sits comfortably on their dorsum, which is their back, essentially. The wire is stuck on using silver glue, and again, it's all about using things that are conductive because it's an electrical circuit. And so the insect is connected to this electrode, which is its gold wire. It's placed onto the leaf, and then it's actually able to walk around a certain amount and select a feeding site. And we want them to be able to walk, to touch the parts of the leaf with their antennae and with their mouth parts, because it takes a wee while for them to select a feeding site, decide they're going to feed, and accept that plant as something they want to feed on. So this setup is designed just to compare plants with differing levels of the bioactive compounds in their leaves.
Yes. So trying to find out what grape plants have more defensive compounds in them and as a result are less tasty to the bugs.
Exactly. So there's some work that was done before I started my PhD looking at mealybug preferences on different grapevine varieties. And one of the things that was found was that some grapevines certainly are less palatable to mealybugs than others, and also that there's a class of bioactive compounds that correlated with that feeding deterrence. So I'm really interested in those bioactive compounds. And so grapevines that produce naturally higher levels of those bioactive compounds, if we can find out whether the mealybugs really don't like feeding on grapevines with those compounds in them, and then breed for enhanced levels of those compounds, then we can protect our vineyards in that way.
So what does a normal day for you entail?
To set up these experiments, I go and collect plant material from usually the glasshouse, and I set up my experiment with the plant material, apply the tiny voltage to the plant and get that done first. And that's important because I work with plants and I work with mealybugs. I want to always work with plant material first so that I don't accidentally infest my plants with mealybugs. And then I will go and collect the mealybugs, and the mealybugs are kept in a separate location. I maintain a colony of mealybugs on sprouted potato tubers. They quite like the sprouts of potato tubers. So I'll collect the insects that I'd like to use, and I have to carefully brush them to allow them time to remove their stylet from the tissues that they're feeding on so that I don't harm the insects. They're very gently moved with a paintbrush. I bring the insects back here, I clean them, attach them to the electrodes and set up this equipment. And that process does take a couple of hours just because attaching these insects to the wires is probably the most time consuming bit, and it's really important to be gentle with the insects and allow them a little time to just recover from being handled by me. Yeah.
So once the bugs are connected, you can watch their eating waveforms on a computer screen between the two cages.
Yes, and I don't have to sit and watch the data for all of the 48 hours, thankfully, because it will record all of the data. I normally do stay for at least maybe 10 minutes, and what I'm making sure is that the insects are able to walk around and that ideally they might even initiate a probe quite quickly.
Do you get kind of attached to some of these insects because you spend so much time looking at them and studying them?
I quite like mealybugs. I think they are very cool looking. It's a good thing I like them because I do look at them quite a bit. There was a time when I was starting to learn how to clean and attach the wire to these insects where I would occasionally name them because they were my little pets, really. However, I've looked at hundreds of these things. So, yes, I can, even though I know that they're a pest, I do like them. Yeah.
What sort of names did you give the mealybugs when you first started this research?
So when I very first started, I was using the adult females. So I would use names of my grandparents' generation. So I would give them names that I didn't know any particular young people with the name because that might feel very strange. So, yes.
What are your end goals and how are you going to achieve them?
So, ultimately, what I'm using this for is I want this information on mealybug feeding behaviour to be able to inform breeding of rootstock varieties so that we can breed varieties that are resistant to mealybug feeding and therefore reduce virus transmission in our vineyards.
How long is this program going to go on for?
So I'm doing my PhD. I'm in my second year now. The project is currently set to finish towards the end of next year, and hopefully we get to continue it beyond that. The current project is funded through MBIE and through the Endeavour Fund and is called Smart Adaptive Rootstocks for a Changing World, and funding for that part of the project is currently set to end of next year.
PhD student Michelle Thompson talking to Cosmo in her lab at Plant and Food Research in Lincoln, now part of the Bioeconomy Science Institute, which also includes ag research, Manaaki Whenua land care research and Scion. Check out the RNZ website, rnz.co.nz, to see some amazing photos of the mealybug attached to a gold wire.